Hello and welcome to the video. Today we are going to go over mitosis. Mitosis is the process by which an average cell in your body replicates itself so that older worn out cells can be repurposed and replaced with new ones. Just to clarify, this is going to be mitosis for most eukaryotic organisms, which are generally multicelled and have organelles and nucleuses, as opposed to prokaryotes, which don't have organelles or nucleuses. Alright, before we begin, we're going to go through an overview of what happens during mitosis. Mitosis has 4 basic stages: prophase, metaphase, anaphase, and telophase. Sometimes, though, people will split prophase into 2 phases, namely into prophase, also known as early phase, or prometaphase, or late phase. There is also another important part called cytokinesis, which starts either during anaphase or telophase, but I will explain more about that when we get there. First, let's start with how the cell looks right before prophase. This state is called late G2 phase. Inside the nuclear membrane, also called the nuclear envelope, the cell has already copied the DNA so that the chromosomes in the nucleus each consist of 2 connected copies, which are called sister chromatids, or just chromatids for short. Animal cells develop 2 spindly structures called centrosomes, whereas plant cells use a different type of microtubule organizing center that does basically the same thing. In fact, mitosis looks almost exactly the same for plant cells as for animal cells, except for one other difference, which I will get into later. But for now, let's go ahead and start right into prophase. In the first part of prophase, early prophase, many things happen. First, the chromatids start to condense, which makes them easier to pull apart. Second, the mitotic spindles begin to form between the centrosomes as they move apart. The spindle is made of strong fibers called microtubules. Lastly, the nucleus where ribosomes are made disappears and the nuclear envelope starts to break down. In prometaphase, or late prophase, the nuclear envelope begins to break down. The chromosomes finish condensing, and the mitotic spindles grow more and begin to grab onto the chromatids. Next comes metaphase. During this phase, the spindle has gotten hold of the chromosomes and have aligned them along what is called the metaphase plate. This plate doesn't actually exist, but rather is a term to describe the plane where the chromosomes line up. The microtubules of the spindles attach to the kinetochores of each sister chromatid pair. Basically, kinetochores are just that spot on the chromatids where that attachment takes place. Before continuing to the next step, the cell will double check that the chromatids are lined up correctly along the metaphase plate, and that the spindles are correctly attached to the kinetochores. This check is called the spindle checkpoint, and it helps make sure that replication goes correctly. Alright, that's metaphase, and we're already halfway done with mitosis. The next step we're moving on to is going to be anaphase. In this phase, the sister chromatids will separate as the protein that held them together breaks down. As they are pulled towards opposite ends of the cell, the sister chromatids become chromosomes in their own right. This stretching happens via microtubules that are not attached to chromosomes, but rather instead push off each other in order to elongate. Alright, that's anaphase, so let's go ahead and move right on to telophase. During this phase, the mitotic spindle breaks down. The nuclear membranes reform and the nucleus reappears. Lastly, the chromosomes begin to decondense back into their stringier forms. Alright, so that's basically mitosis, but one more thing that's important to talk about before we end completely is cytokinesis. So let's go ahead and do that. As I said before, cytokinesis either starts during anaphase or telophase, depending on the cell. Then it will finish just after telophase. Since cytokinesis happens differently in animal and plant cells, I have made up an example of both for us to compare. In an animal cell, the cellular membrane of the splitting cell is drawn together until that one cell becomes two. This pinched crease is called a cleavage furrow. Plant cells, however, can't be pinched together like animal cells because they have a solid structure called a cell wall, which is not flexible enough for that kind of pinching together. So instead, the plant cell forms what is called a cell plate down the middle of the cell. This eventually comes together to separate the cell into two new cells by forming a cell wall down the middle. Well, that is mitosis. I hope you enjoyed this video and found it helpful. Have a great day. Bye.